Welcome to Quantitative Analysis, Inferential Statistics, Part 2. We've already discussed the types of statistics. Our first video was related to descriptive statistics. We soon will have one on correlational, and video 2 and video 3 will be on inferential statistics. These are the most powerful statistics, and they let us know how much confidence that we have in our ability to generalize our findings of a study to our population of interest. So these are the powerful statistics. The underlying principles of inferential statistics have to do with probability and distribution of findings within a population. So the sampling that we saw that was most powerful is randomized sampling from a population. Likewise, the inferential statistics are dependent on that. So if you're running inferential statistics, but you haven't performed a random sampling, that is a major flaw in a study. We are testing hypothesis with inferential statistics because, remember, we want to identify with these either causal or correlational relationships in our findings. So we use inferential statistics to draw conclusions about that we can generalize to a population. There's two different types. One is hypothesis testing, which we're going to discuss here, and the other is parameter estimations or estimating for populations, which we'll be talking about with confidence intervals and odds ratios coming up. So this has to do with theoretical distributions of the sample related to the population. And a distribution is an important core concept that we are examining with inferential statistics. So let's start off with p-values. P-values are basically letting us know if our findings are in fact statistically significant. So what's the probability that there's no difference in our sample, basically our experimental sample and the population, no relationship between the two, no prediction, and that what we see is just what's expected. So because of this, the smaller the number, the p-value generally, let's say 0.05 would mean that 5 people out of this sample that we've drawn, the finding that we have is actually just due to chance and not due to the effect. So 5 out of 100, that's not very many. And so the p-value is a number we want as low as possible. So we want that number to be very small. Here's the statistical definition of the p-value. The chance that the result observed would occur if the null hypothesis was correct. Remember our previous slide? That has to do with no differences. Those are all the null, means no hypothesis, no difference, no relationship, not predictive. So this is the possibility, the chance that the results we see would occur if there was nothing going on, if the null hypothesis were correct. So here's an important point that the general public doesn't understand, and that is that the statistical significant differences are not necessarily clinically significant. So if there's small differences, does the researcher mention this, and are they clinically meaningful? So remember, when we're looking at statistics, we're still storytelling. We're just storytelling with numbers. Statistical significance is the least interesting thing about the results. This needs to be described in terms of the measure of magnitude, not just does a treatment affect people, but how much does it affect them. We see this problem in diabetes management because it is very easy to reduce the A1C of people who have very high hemoglobin A1C. So when we sample people and include them in a study, if they're included in a study, if they simply have an elevated A1C, like in the magnitude of a 10% A1C, then it's very easy to drop that by 1%. it's much more interesting if we can take someone with a moderately high A1C, like 8%, and drop that down to a normal value. And so always we have to consider the effect and the effect size or the power of the observation to show a meaningful difference. So this is the mathematical equation for effect size. You're not responsible for knowing that, but it's the experimental group minus the control over the standard deviation. So this difference that observed should be a meaningful difference, and to be meaningful, it might be small or it might be a large effect. And that should be something that when you read research, which is what we're focusing on here, when you read research, you want to see the write-up of the report reporting on the effect size. So then here's the important piece for applying any of these statistics. You must have a sampling distribution around the mean. So this is an important assumption of sampling, and that's that there's a normal distribution and that the mean of the sample equals the population mean. The standard deviation is called the standard error of the mean, the way that the data approach this central measure. So we estimate using a point estimation. So it's a descriptive statistic that estimates the population value. We're going to talk about this in the next presentation more. This is the odds ratio, also abbreviated OR, and we could use means and percentages. So these are all estimation parameters that we use because we cannot sample an entire population. So we have to use a mathematical estimator to determine what the mean, based on the sample that we have, what the mean of the population is. And then we have interval estimation, and this is a range in which we think the population value probably lies. An interval estimation is called a confidence interval. And so remember, point estimator or an odds ratio. So that's a very specific number like 5 out of 100 or a mean point. Think of it as you can point to it. You can point to a mean, point to a percentage, point to an odds ratio. An interval is another term for like a range. So we have a confidence interval. So that's the range of numbers where we're confident that the population value lies in that interval. So they indicate the upper and lower confidence of the population values between those two limits. So this is how you read this in a study. 95% confidence interval of 40 to 50 for a sample mean of 45 indicates there's a 95% probability that the population mean is between 40 and 50. So this again gives us an idea of what that is and how a way to interpret those statistics. So let's look to the outcomes of statistical decision making. You will see there's type 1 and type 2 errors. So this is something you need to understand the kinds of problems that come. So if we accept the null hypothesis, which is true, and the actual situation is the null hypothesis is true, then that's a correct decision. Remember, this would be a study that shows no difference between the intervention group and the regular population. So that the null hypothesis is accepted, that there's no difference. And so the null hypothesis is true. Then look down here. Here's the other thing that would be true, that the null hypothesis is rejected. When a null hypothesis is rejected, that means that the intervention had an effect. And if it's rejected, then those are both correct decisions. However, when you instead say that there is a difference here, that the null hypothesis is rejected, and in fact, the null hypothesis should not have been rejected. There's nothing happening. Remember, this is true, that a null hypothesis is true, means nothing's happening. When it's false, it means there's something happening. That is a type 1 error. So when you think that there's nothing happening, but there was something happening, that's a type 1 error. And the opposite is also true. If you think that there really is something, but there was nothing happening, then that's a type 2 error. Using this chart and understanding how this is set up can help you understand the kinds of errors that can occur with statistical modeling. I'm briefly going to mention power. We've talked about it before. Power automatically increases as the number of participants increase. Usually they do a power analysis based on the amount of difference the researcher deems is significant. This is a clinically significant amount of difference. In my example with A1C, I would say that a 0.5 to 1% change in the A1C in a population of people with an 8 or lower would be significant. So I would start there and then ask how many people I need to sample to detect if that is true. And that's what power means. All right, here we are looking at statistical tests for hypothesis testing. So when we look at a t-test, we're looking at the difference between two means. When we look at ANOVA, it's 2 or more means. A regression tests a relationship, and so does a logistic regression, and a chi-square is used to look at the relationships categorically. Here's where our understanding of level of measurement becomes so important, because if you have categorical data, you cannot perform these two tests. These tests have to do with means, and to have means, you need to use interval data. On the other hand, regression can be done between those both continuous and interval variables, but it has to do with the type of sampling that was done. And a chi-square can only be, or you would run that because you only have categorical data. You want to use the highest test available to you based on your procedure. However, even if you have wonderfully sampled data that's randomized to the procedure, if the data itself collected is categorical, the highest statistical test you can use is a chi-square. Here's the similarities between t-test and ANOVA. Notice difference between two means, difference between 2 or more means. In both cases, the dependent variable can be continuous or interval, and the independent variable, the variable that is causing the effect, can be categorical, however. So as long as your dependent variable, your outcome is continuous or interval, you can run a t-test or an ANOVA. And there's various types of them. You're not going to be responsible for knowing those various types. But do know that the whole point of t-test is to compare means. So here we have a control group and a treatment group mean. And interestingly, they overlap in this range. When the two means do not overlap, then we know that there's most certainly a big effect. But when they do have an overlap between the control group and a treatment group, it means that we have some confidence that something was happening in the treatment group, but not as strong as we would like. This symbol, this M, is a symbol for mean. So when we compare group means in ANOVA, we could have 2 or more groups, and we look at all these different means. In this case, it would be a rather wide range that the population is occurring in, because all of these do not, in fact, overlap. When we look at a regression, what we're looking at is continuous independent variable and dependent variable. So here we have income and health risk, and what we're looking for is how much do these two values have a relationship that goes in a line. It would be you'd be hard pressed if this line was not already written here to know how to draw it. You might draw it straight through, but because of the statistical analysis, we can see that there's an inverse relationship, which means as health risk declines, so does family income. And family income increases, I mean. So the relationship between the independent variable and the dependent variable here is negative, inverse, and here it's considered positive. So as these behaviors increase, so do the number of educational years. So there's a positive relationship between these two variables and a negative relationship between health risk and family income. So as family income increases, health risk also decreases. So we use a regression when we want to ask if one thing can predict an outcome. So it's all done by the slope of the line. So here we have an example of a prediction. Does high school GPA predict SAT scores? Mathematically, this is the formula for the slope of the line. And does birth weight and height predict adult BMI? So there's an example of a type of question that's asked of a regression. Note, a regression is asking about prediction. It's not saying that one causes the other. It's not a causal relationship. It's simply a relationship between two measures. So as the change here, you put the dependent variable, and then you have one change in x, which is the independent variable. So run your dependent variable here and your outcome on the base of the line to observe your regression. Here's an example of that every 5-point increase in the brain scale, the risk for hospital-acquired pressure ulcers at 1 week decreased by 36%. So this is our outcome variable. This is our predictor variable. And so this would be an inverse or a negative relationship. The chi-square test looks at two categorical values, and so you're observing in this case the frequencies in each cell, and then what you would expect if there was no relationship versus finding a relationship. So it's about the goodness of fit, observed minus affected squared over expected. So that's the sum of these equals the chi-square findings. So the major takeaway from this part 2 is that you should know the difference between statistically significant and clinical significance. And in the research you're reading, the author should discuss these two things. And as you critically read something, you should think about if the finding is in fact clinically significant. You need to know the difference between a type 1 and a type 2 error, and know the difference between hypothesis testing statistics and the one that you can use based on the level of variable. And if you are testing the means, differences between means, the relationship between variables, or frequency. Remember, frequency has to do with chi-square, categorical relationship, categorical variables. A relationship can have it may use an interval related data, but what you're looking at is a cause and effect. And then, or really not cause and effect, but a predictor, a precursor and an outcome. And means testing then is our strongest, and it looks at the means in the treatment group versus the population mean to determine if there was a statistical effect. So these are the major takeaways of this 20-minute video. Thanks for joining us.